Hi everyone, welcome to Medical Microbiology. So today we will discuss chapter 14 and 15. So these two chapters talk about human defense mechanisms. Therefore, I combine these two chapters together in one lecture. Okay, first of all, we have to understand our natural defense mechanisms, which is consists of white blood cell. Okay, so this is what we call the armies, just like the armies in our country defense mechanisms. So this is the people, the armies that uh, fight against infections. There are five types of armies or white blood cells in our system. The first one is called the neutrophils. These neutrophils are the white blood cells that usually fight against bacteria. The second one is called the lymphocytes, and this second type of armies, they usually fight against viruses. And then the third type of white blood cell is called the monocyte, or sometimes it is called also macrophages because they are very big white blood cell, and they usually also fight against bacteria. The next one is eosinophils. The eosinophils, they usually fight against parasites like worms and protozoa. And then the last one is called the basophils, and they usually fight against allergens, and they produce what we call the allergic reaction. So these are the five types of white blood cells. Okay. I put this uh, list is in sequence, which is from the most abundant ones to the least abundant one. So neutrophil is actually the most abundant type of white blood cell in our system. The lymphocytes going to be the second abundance type of white blood cell, and then monocyte, eosinophil, and basophil. So basophil is the least abundant type of white blood cells in our system. In addition to the army, in our body, we also produce the weapon. So remember, the weapon is actually produced by this army. So there are some army that produce special weapons. Okay. The main weapons is called the antibody. Okay, so the antibodies is actually the main weapons in our body. And these antibodies, they are produced by the lymphocyte, especially the B cell. So lymphocyte consists of two types of cell, B cell and T cell. So these B cells are the one that produce this antibody. Okay, there are five types of antibodies in our system. Okay? Sometimes the antibodies is also known as immunoglobulin or IG. That's another name for antibody, IG or immunoglobulin. So there are five types of antibodies. The first one is called the IgG. That's the most abundant one. Okay. The second one is called the IgA. The next one is called the IgM. And the next one is called the IgD. And the least abundant one is called the IgE. Okay, so again, these weapons or the antibodies is produced by the B cell. These antibodies is also fight against some specific infection. For example, IgG mostly fight against any type of infections, okay, whether it is coming from bacteria, viruses, parasites, fungi, and etc. Okay. The IgA, this is also fight against infection, all type of infections. However, this IgA usually located in the fluid, not really in the blood. So this IgA located in the fluid that we produce, like for example, in the tears, mucus, sweat, there are IgA in this fluid. Okay, so IgG located in the blood, while the IgA mostly located in the secreted fluid. The IgM, okay, this is the next abundant ones in the blood. They usually fight against foreign cells. For example, if we have blood transfusion, so we receive the cells that come from other people, and this cell is considered as a foreign cell, and usually the IgM will try to fight against this type of antigen or foreign cell. The IgD, they usually have other antibodies or either other immunoglobulins. And the last one, the IgE, the least abundant one, this is the one that usually also help to fight against allergen. It is kind of work together with the basophil. You see the basophil and IgE, they usually fight against allergens, the antigen that cause allergy. Okay. There is additional weapon that also produce in our system. Okay. The first one is called the interferons. This interferon is usually produced by the infected cell. So if we have the cell that is infected by viruses, for example, this infected cell able to produce interferons. And these interferons then will go to the healthy cell, try to give warning to the healthy cells about the virus or about the pathogenic microbes that enter into our system. The next one is a proteins. A group of protein is called the complement system, and this usually fights against bacteria. So help us to fight against bacteria. And the last one is toxin. This toxin will also fight mostly against viruses. Okay, so remember the antibodies, the 5 type antibodies is produced by the B cell. The toxin mostly produced by the T cell. So this T cell produce the toxin that will destroy the viruses. The way they destroy the virus actually by destroying the infected cell. So this toxin also produced by the T cell, it also will destroy the cancer cell. So the cancer cell usually fought by the T cell. It's destroyed by the T cell by producing the toxin. Okay, again, we review the armies, which is the white blood cell. Again, there are 5 types of white blood cell. The most abundant one is called a neutrophil. About 55 to 70% in our blood. Okay, so it is a neutrophil. These neutrophils is used to fight against bacteria. So when the neutrophil increase in number, for example, you take the sample, the blood sample, and calculate the neutrophil, lymphocyte, monocyte, eosinophil, basophils. So the natural ratio is between 55 to 70%. However, if from your sample, for example, this neutrophil increase into 90%, then it will give us the indication that there is a bacterial infections in our system. Okay, so it can be used for diagnose infection. Okay, the second type of white blood cell is called the lymphocyte. This is the second most abundant type of white blood cell. And again, the lymphocyte consists of B cell and T cell. They usually present in about 20 to 35%. So this is for healthy blood, 20 to 35%. Now, for example, if we take a sample and then lymphocyte is actually ratio actually increase, let's say into 50%, then it is indication that there is a viral infection. Because what? Because this lymphocyte fight against viruses. So increase the number of lymphocytes give us indication that there is a viral infection. The next one is called the monocyte or macrophages. This is the one that also fight against bacteria. And the normal ratio is between 3 to 7%. Again, if we take a sample, blood sample, and calculate them, let's say increase to 20%, this is indication that there is a prolonged bacterial infection. Okay, and the next one is called the eosinophil. The normal concentration is around 1 to 3%. And these eosinophil mostly fight against parasites like worms or protozoa. Now, if we take a sample again and then we calculate and this eosinophil increase, let's say, into 10%, then it gives us the indication that there is a parasite in our body. Okay, so this is how we use this differential calculation of white blood cell to diagnose disease. The last one is called the basophil, and this is very, very low concentration, less than 1% in our blood, and they usually use for fight against allergen, the antigen that causing allergic reaction. If, for example, in our sample we calculate and this basophil increase to 10%, then it is an indication that there is allergy or allergic reaction. So people that have allergy, they usually have increased the basophil concentrations in their blood during the allergy season. Okay, so these are the main army that fight against infection or disease that present in our body. The study of body or human defense mechanism is called the immunology, especially when we study the lymphocyte over here, okay, which is the B cell and the T cell action. Okay, so this is called the immunology, the special scientific study of immune system. So for the healthy humans functions, the immune system usually have three main action. First, they usually survey. They always survey our body. So this is what we call the surveillance. So like a T cell, they usually go around our body using the blood circulation and checking if there is something happen in our body. Like for example, if there is cancer cell developed, then the T cell will destroy that cancer cell as soon as possible. So this is what we call the surveillance. So that's the first action of healthy immune system. And then when they survey, then they might recognize if there is a foreign material. This foreign material is also known as antigens. So they will recognize it. And then these antigens or this foreign material then will be destroyed by our immune system. So this is what happened in our body. It's always happening, which is the immune system will survey our body, our system. If there is foreigners, if there is antigens, then they will destroy that antigen. Okay? They will recognize it and then they will destroy it. And when the antigen destroy, then we become, we keep our body healthy. But if we are unable to destroy that antigen, then we have the disease. So sometimes we have to take drugs from outside, like antibiotics, in order to help our immune system to fight against these stubborn antigens, like maybe bacteria or viruses. So antigens can be anything that actually causing disease in our body, provoke the immune system in our body. That's what we call the antigens. It might be bacteria, viruses, allergens, worms, parasites, or even sometimes our own cell become antigens in our body and causing the disease. So the disease that caused by our own cell, our own antigens that coming from our own body, it is called the autoimmune disease. So the disease that caused by our own cell. Okay, again, this is the process. So the immune system, especially the T cell, will always perform the surveillance. Okay, surveillance. So, okay, if we talk about our community, maybe this T cell, they work as like a sheriff. Okay, like a sheriff or police that always survey the neighborhood, check if there is something happening. So this sheriff or this white blood cell will check if something happened or not. If there is nothing happen, so they check this normal cell, and then nothing happened, and say, okay, there will be no reaction. You okay? You fine? Okay? And then this white blood cell then will survey other area. So just always like that, do the surveillance. If, for example, okay, this white blood cell see that this one is not our own cell, so this not our own cell will give special markers over here, and then this white blood cell, okay, for example, the T cell will recognize this foreigner. Oh, this is foreigner. And then if it is foreigner, then this white blood cell will destroy this foreigner. So destruction. Okay, so this is what happenings in our body over and over. Surveillance, then recognition. If it is just our own marker, then there will be no reaction because this white blood cell will not kill our own cell. This white blood cell will not destroy our own cell. But if this one is foreigner, so it will be recognized with the white blood cell. Why it is able to recognize? Because the marker will be different over here. Okay, so the markers of the foreigner cell will be different than the marker of our own cell. So this one, the cell will be able to recognize this foreigner, and finally they will destroy this foreigner. Okay. And if, again, if we're unable to destroy these foreigners, then we have the disease, and sometimes we will have to take medicines or drugs from outside in order to destroy this foreigner or this disease, the pathogens. Okay. Okay, based on that, then we can actually classify our host defense mechanism. So our defense mechanism, this is human defense mechanism, can be divided into two main types. The first one is called the non-specific or innate. So what is non-specific mean? It means that these defense mechanisms will fight any type of infection. It is not specific. So any type of infection, any type of infection. Okay, so it fights against any type of infection. This is what we call the non-specific. Innate, so innate means, okay, we have it since we were born. So we have this type of immune or defense mechanism with us since we were born. So we born with this non-specific defense mechanism. Okay. The second one is called specific or acquired. Okay. So specific means it's only fight against specific infection. Okay. For example, there is a specific defense mechanism that only fights against chickenpox. It cannot be used to fight against polio, as it's only one type of infection. Fight against one type of infection. Okay, so one type of defense mechanism fight one type of specific infection. So we have several type of this specific uh, defense mechanism. And these ones acquired. It means that it has to be made. Okay, it has to be made. It is made when we have infection or the disease. Okay, so compare with this innate over here. So we have it since we are born. We're born with this innate defense mechanism. So we have it whether there's infection or not. We have this type of defense mechanism, which is non-specific and innate. For the specific ones, we only made this type of defense mechanism whenever we have the infection. Okay, for example, we have a chickenpox infection, then we will make the defense mechanism that will fight only against chickenpox infection. Okay, now the non-specific defense mechanisms can be divided into first line of defense and the second line of defense. The first line is mostly used for prevention. Okay, so prevention or for blocking the entrance of disease, right, or infection. Okay, so this is for blocking or prevention. The second line of infection, yeah, this is happening when the first line of defense mechanism fail, then the disease or the infections or the pathogen will enter into our body. Then the second lines of defense mechanism will try to remove it. Okay, so this is the action, okay, after the infection enter to our sterile tissues. So we have what we call the second line of defense. If the second line of defense fail, then we still have one more line of defense. It's called the third line of defense. And this third line of defense is actually part of the specific defense mechanism. Okay, so this is the process of defense mechanism. Start from the blocking the entrance, which is the first line of defense. Then if it is fail, then it's go to the second line of defense. If the second line fail, then it will go to the third lines of defense mechanisms. Okay, now what are the first line of defense mechanism? Again, this is a prevention, the blocking of the entrance of microorganisms. It can be done physically, okay, by the physical barrier. Like for example, our skin. Okay, our skin is very tough. It has many layers of cell, so it's become physical barrier of infection. Okay. Another example is in our nose, for example. There are hair, there are mucus that actually become physical barrier for entrance of many microorganisms. Okay, so the microorganism will be filtered by this hair and also the mucus in our nose. So this is example of physical barrier of the first line of defense. It can be also performed by the chemicals that we produce. Okay, like for examples, in our sweat, there are enzyme, which is chemical, it's called the lysozyme. And this enzyme is actually able to destroy the microorganism. The sweat itself is physical barrier because when we are sweating, we actually physically remove some bacteria or microorganism. Spit, for example, also physical barrier. Coughing, sneezing, also physical barrier. Tears that we produce, this is also physical barriers, the first line of defense mechanism. But in the tear itself, there is a chemicals. In the sweat, there is a chemical that able to destroy microorganisms. That's become the chemical barrier. Okay, and then the last one for the first line of defense is genetic barrier, which is some people they have special gene that help them to protect against some disease. For example, people that live in malarian belt in Africa, they have special gene that actually protect them against malaria. So if this gene present, then they will be protected. If it is not, then it will not be protected. So this is kind of genetic barrier for some people that have a special genes to fight against specific infection. So this the first line of defense, which is mostly prevention or blocking the entrance of disease. If again the first line of defense fail, then the disease, the infection will enter into our body. Then we have the second line of defense. It starts with the inflammatory responses. Then there will be interferon production, phagocytosis, and also complement reaction. This is part of the second line of defense. We will talk about this one later. Okay. And if the second lines of defense fail, then we still have one more, the third line of defense. And this is type of specific defense mechanism. It is done by the T cell and also by the B cell. Okay. Then we will talk about this third line of defense later. So this is a summary of human defense mechanism, which is divided into two types. The first one is called the non-specific defense mechanism. This type of defense mechanism will fight against any type of infection, and we born with it. Therefore, it's called the innate. And this non-specific defense mechanism consists of two lines, the first line and the second line. And then the second type of defense mechanism is called the specific defense mechanism. It's only fight against specific infection, and it is acquired. It will be made after the infection or during the infection. Okay. So this specific defense mechanism is also become our third line of defense mechanism. Okay. So we start with the non-specific defense mechanism, which is this is actually discussed in chapter 14. So non-specific host defense, and this is also called the innate defense mechanism. We actually born with this defense mechanism. Okay, so the non-specific defense mechanism is also known as innate defense mechanism. Okay, again, it consists of three types. Okay? The first one is called the first line of defense. It consists of physical barrier. And again, we actually have a lot of examples of physical barrier or the first line of the first lines of defense. So this is actually the action that will protect us against the entrance of the disease. Like for example, the skin structure. It has so many layers. So our skin, if it is healthy skin, it will protect us against the entrance of many pathogenic organisms, microorganisms. But whenever there is a break in the skin, maybe there is a cut in the skin, then it is actually the place where the microorganism will enter into our steroids. So make sure when you have a skin cut on the skin or damage on the skin, make sure you protect this, you take care of this damaged skin. Otherwise, it's become the place for the entrance of microorganism into our sterile body, especially to children, because sometimes children just don't care. So as a parent, then you have to take care of this problem. You put something that will protect the skin from infection. Sweat. Okay, when we're sweating, this is also physical barrier, and we actually remove a lot of microorganisms from our body. Mucus, like for example, mucus in our nose, it will bind many microorganisms. It kind of protects them from entering into our sterile tissue. When we blinking our eyes, then produce the tears. That's actually also remove a lot of microorganisms from our eyes. Uh, that's the physical barrier. Uh, I think this is not physical. Stomach acid is not physical because acid is chemical. So we will put that on the next one. The hair, okay, hair in our nose, for example. This is also uh, filtering mechanism, which is physical barrier of our mechanism. Another example is sneezing, okay, coughing, urination, defecations. That's all example of physical barrier of the first line of defense. Because when we sneeze, when we coughing, when we urinate and defecate, is actually remove a lot of microorganisms from our body. So it's kind of protect us, okay, from maybe infection. The second first line of defense is called the chemical barriers. It is done by chemical. Like for example, oil. Okay, so the oils in our skin, they're actually able to kill many type of bacteria. Lysozyme, this is enzymes that we produce and present in the tear, in the sweat. This lysozyme will kill a lot of bacteria. The tear and the sweat itself is part of the physical barriers. But the enzymes that is present in the tears and the sweat is part of the chemical barrier. So it's actually double uh, mechanism, okay, from physical and chemicals at the same time. High lactic acid and electrolyte concentration in the sweat, in the tear, they're also able to kill bacteria. Skin sometimes it has acidic pH, uh, hydrochloric acid in the stomach. So this is also chemical. There are so many bacteria that will enter into our stomach through the food and the drinks. And because stomach is very, very acidic, it actually kills a lot of bacteria or viruses that coming from outside into our GI tract. Okay, so this is very important over here. Okay, the hydrochloric acid in the stomach. The digestive juices, enzymes, and bile in our intestine is also able to destroy some type of microorganism. Cements also contains some antimicrobial chemical that will destroy some uh, microorganisms. The vagina also have the acidic pH, which is also help that structure to kill several type of microorganisms. So this is chemical barrier. Okay, so I think should be one more the genetic barrier. So the chemical barrier, I believe in the textbook is mentioned about genetic barrier. Okay, so example, some people that live in the malarian belts in Africa, they have special gene that able to protect them against malaria infection. Okay, this is the picture of some first line of defense, like for example, the skin structure. Okay, the skin structure, the sweat glands. Okay, production of sweat and lysozyme at the same time, wax in our ear, and also protection against some entrance of low pH in our skin. Cilia, this is a physical barrier in our respiratory tract. So the cilia actually remove many bacteria from lower respiratory tract into our mouth. Saliva is also have lysozymes on it. Mucus in the nose, tears, and then oil glands in our skin. Urination, defecation, that's also part of the first line of defense mechanism. Stomach acidity, this is also part of the first line of defense mechanism. So we actually have this type of defense mechanism even before the infection. So this is the protection, this is the blockage against the entrance of microorganisms that might cause disease to us. Yeah, this is the third one over here, the genetic barrier. C. Okay, so again, some people have the ability to protect themselves against disease based on their genetics differences. Okay, that maybe exists in some community. Okay. So examples is people that live in the Malarian belt country in Africa, they have special gene, it's called the sickle cell gene. Okay, so let's say normal gene is like this. Let's say normal people carry this type of gene, normal. So they usually normal. Okay, and then people with the sickle cell gene maybe carry this. There's a sickle cell, so they this person actually will have what we call the sickle cell disease, which is a type of anemia. Okay, but if they combine, let's say like this, and this one is heterozygote, this person usually able to is protected against malaria. Okay. So people that have normal gene usually is not protected. Again, it's not protected against malaria. So they will usually maybe die because of the malaria. So most people in Malarian belt country, they carry this type of gene, which is the heterozygote type. So when they move to United States, for example, okay, some of them maybe, let's say this one married with another one that carry heterozygous gene, then they might have children that carry both of the sickle cell gene. So when these children have both sickle cell gene, then these children will have disease called the sickle cell anemia. Okay, so mostly the sickle cell anemia present in the African American people because they are, their parents or their forefathers that coming from African country carry this sickle cell gene with them. Okay, so we're done with the first line of defense. Now we go to the second line of defense. Okay, so the second line of defense, it consists of several actions. Start with the, of course, recognition. Right? So recognition, so when the antigens, when the pathogenic disease or infection coming into our system, they will carry special markers, and then our immune system will be able to recognize it. And then after that, yeah, start the first process is called the inflammation. So whenever we have, for example, damage or infection, that site of infection usually become inflamed because this is actually the first response of the second line of defense, which is the inflammatory responses or inflammation. Sometimes it's also accompanied with fever, okay, higher body temperature. The next process will be phagocytosis, okay, which is uh, distractions of pathogens by phagocytes or cell eaters, the cell that able to eat pathogen. It's called the phagocyte, and the process is called a phagocytosis. And also our cell will produce the interferons. Usually the infected cell will produce interferon that will give warning to the healthy cell. And also our cell will produce what we call the complement system that will destroy the pathogen. And we'll see one by one the process. Again, the recognition, it is done by mostly the T cell. Okay, let's say this is a T cell. So this T cell actually have special proteins that able to recognize the foreigner cell. Okay, let's say this like this. So this is the proteins that able to perform recognition. Let's say this is foreigner cell, or like maybe uh, pathogens, or some mostly it's called the antigens. Okay, so anything that provoke the infection or immune system. This one will carry special sign or marker. Okay, that will be recognized by this receptor cell. So this one receptor. Okay, so the T cell have receptor that will recognize the marker. So when it is recognized, then we know there is a problem. Okay, so this is the first step, which is recognition. And this then go to the second step. Okay, second action, which is the inflammatory responses. So whenever we have infection, we will have this response. It's called the inflammatory responses or inflammation. There are several signs and symptoms of the inflammatory response. Of course, redness. To the area of infection, they usually become more red. This is what we call the redness. Why? Because this area that have infection usually will receive more blood, because blood is the one that carry our immune system, the one that carry white blood cells, eh? Neutrophils, lymphocyte, monocyte, they all carry by the blood. So therefore, when the blood come into this inflammation area, that place become red. So this is what we call the redness. And that area also become warmer, okay, warms, because the action that happening in that area where the white blood cell try to destroy the pathogens, the antigens. And then it will be swelling, okay, inflamed. This is the area become bigger. So this is also the sign of inflammation. Okay, warm, redness, swelling, pain. Okay, this is the symptom, which is some when we have an infection. Sometimes in that place we feel the pain or maybe itch, and there is a possible loss of function on that side. So that those are the sign and symptom of inflammation, which is the second response of second lines of defense. Again, this is just examples when our tissue get injured, boom, the pathogens, bacteria will be enter over here, and then our blood cell will carry a lot of blood, white blood cell into the area, then it's become redness, okay, become red, and then it's also inflamed. Okay, so this is white blood cell over here. They fight this bacteria. And so when they're able to fix the problem, then what happens? We mostly have what we call the scar. So scar formation over here after these white blood cell able to fight against this infection. Okay, so this is several action that happenings when we get the infection in specific site of our body. Now the leukocyte, which is the white blood cell, so this is another name of white blood cell is leukocyte. So some white blood cells have a unique characteristic. Okay? For example, they're able to move from blood circulation into the damaged tissue. So remember, they are carried by the blood, but they're able to move out from the blood vessel. This is what we call the diapedesis. So the action is called the diapedesis. And they also have the ability to move to another side based on chemical sign. Okay? So chemotaxis is mean that the movement due to the chemical sign. So the white blood cell able to migrate or to move to certain area when they sense chemicals on that area that maybe is not present during normal time. So this is what we call the chemotaxis. So they're able to move based on the chemical clue, and they're also able to move out from the blood vessel into the infection site. Okay, so this is examples of diapedesis. So the white blood cell carried by this blood vessel. Okay, they able to actually move from this white blood vessel out. Okay, so move out from the water. They're able to squeeze, squeeze between this epithelial tissue of the blood vessels. And they usually move to the infection area, where the infection area actually give the clue by producing the chemicals. So remember last time we talked about the bacteria or pathogens, they produce the virulent factors like enzymes and toxins. Okay, these are actually chemicals. So these chemicals give the clue for our white blood cell to come to this area and fight them, fight the infection. Okay, the next one is fever. So this is also the second part of the second line of defense. Usually when we have infection, then we also have fever. So this is caused by the pyrogens that may be produced by the pathogens, and this pathogen actually will set our thermometer in our brain. So there is a special area in the brain that set our body temperature. When the pathogens enter into the setting place, then it's increase the body temperature. So yes, we have what we call the fever. So fever actually have benefits from us, okay, which is it will inhibit the multiplication of some bacteria that sensitive to temperature. Okay, so it increase also metabolic rate because increased temperature will increase the metabolism. Okay, so that's the benefit of fever. So if the fever is 101, for example, or maybe 100, that will be still a good fever. So this is benefit us. But if the fever increase to 104, 105, that will be dangerous. So we have to take the fever reducer when it's increased so high. But again, just remember, fever is actually part of our defense mechanism because they're able to kill some bacteria that's sensitive to high temperature, and also increase the metabolisms, the fight of white blood cell in the infection area. The next process of second line of defense is called the phagocytosis. That's the process called. It is performed by phagocyte. Cyte means cell, phago means food or eat. So phagocyte is the cell that eat bacteria. We have three types of phagocyte in our body, which is actually our white blood cell. The first one is called the neutrophil, which is the most abundant type of white blood cell. So they also able to eat bacteria. The second one, eosinophil, they able to perform the eat against the parasite. Okay, and then macrophages, this is the big eater. Okay, this is the biggest eaters. This is the one that actually able to eat so many bacteria in one time. So they kind of clean up the area of infection by eating the bacteria. So these are the three types of phagocyte in our body. And the process of eating of bacteria is called the phagocytosis. When you see the cis over here is actually mean process. Okay. Yeah, this is the process is called the phagocytosis. Okay. This is general activities of phagocytosis. First is survey the tissue that complements, they will recognize, there is a recognition over here. If the tissue of the foreigner cell coming, then they will know because they do the survey recognizing, and then they will ingest it, that foreigner, and then they will extract some information that coming from that foreigner. So immunogenic information that can be used later for us to even destroy more bacteria or pathogen in our body. Okay, let's say this one's here. So this is our phagocyte, which is white blood cell. Yeah. Phagocyte example is macrophages or the neutrophil. And this over here is bacteria. Okay, they are small. So this phagocyte, so you make it bigger over here, able to recognize this bacteria. Okay, and then it will be poo, making this mouth structure, and then eat. When it is eaten, then it will be entered into the cytoplasm, and in the cytoplasm, this uh, vesicle, this phagocyte will produce the lysozyme, and this lysozyme will destroy this bacteria. And this one is bacteria being destroyed, and finally, gone, is died. And this can be removed from the cell. So this is how this bacteria able to eat and remove, I mean the phagocytes able to eat, destroy, and remove the bacteria from our body, from our system. The next ones, okay, the next process in the second line of defense is called the interferon production. So this is actually a type of proteins that produced by the infected cell. So we're gonna see the process over here. Okay, let's say this is our cell that is being infected by viruses. So this is infected cell. This infected cell are able to produce the interferon, and the interferon will be released into the healthy cell. So this is healthy cell, and whenever the healthy cell receive the information, the interferon, then it will make a protection against the infection that come into this cell. So it's preparing. Okay, just this infected cell, the interferon is just like a good neighbor. So when the good neighbor, for example, being robbed, then he will call the neighbor, his neighbor, and try to give the information. Try to warn them, hey, I just get robbed. So make sure you lock, you close your door, the windows, and etc. So this is the interferon, just like a good neighbor that give information to the other neighbor when they get the infection. Okay, the last one for the second line of defense is called the complement system. It consists of about 26 proteins that we produce, and these proteins actually will destroy the bacteria. Let's say this is bacteria. This 26 protein will attack this bacteria and then make a hole, okay, on the cell wall. So when the bacteria cell walls destroy, okay, become the hole over here, then the water will enter into the cell. When the water enter into the cell, then the cell become bigger and finally will be exploded, okay, because of this process. So this is the process of destruction of bacterial cell by the complement system. So this is the process. The proteins attached to bacterial cell walls, 26 or 20 of them, and then making the holes, okay? And then water will enter into the cell, and finally the bacterial cell become bigger, 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 and then boom, it's gonna be explode. Good, this bacteria will destroy. Okay, so that's chapter 14. In chapter 15, we actually talk about the third line of defense. It's called the specific immunity, which is become the third line of host human defense mechanism. Because this is specific immunity, it will be done by specific cell. So that specific cells are the lymphocyte. Lymphocyte, which is the B and T cell. Okay, so B cell and T cell. This is the reason why Another name of this specific immunity is called the lymphatic system or immune system. Because this is done by the lymphocyte, which is the B cell and T cell. Okay, B lymphocyte and T lymphocyte. So this B cell and T cell is not only protect us, it's not only destroy the infection, special or specific infection, it's called the specificity, but it's also produce what we call the memory cell. So whenever we have the infection, special infection, specific infection like chickenpox, for example, then we fight them, and then after we fight them, we have the memory that will protect us against the second infection of chickenpox. So we become immune. Okay, we become immune to that specific disease. So this is the specialty of the third line of defense, which is the B and T cell. Is not only fight against specific infection, but also produce the immunity. Okay, so we become immune against that specific infection. Okay, now the B cell, both of the B cell and the T cell, they are produced in the same organ in our body. It's called the red bone marrow. So red bone marrow is the place for B cell and T cells. The red bone marrow is actually the place for production of all blood cells, including white blood cell, the red blood cell, and the platelets. And remember, white blood cell consists of 5 types: neutrophil, lymphocyte, monocyte, eosinophil, basophil. eh? And then lymphocytes, they have 2 types, which is B cell and T cell. So that's the reason why these cells are produced by the red bone marrow. When the B cell is produced, then it will stay in the red bone marrow to get mature. So it's become mature B cell in the red bone marrow. The T cell over here will move to the thymus gland to be mature. Okay, this is the mature T cell. Okay, just for your information, both of them are produced in red bone marrow, but B cell will be mature in the red bone marrow. The T cell will be mature in the thymus gland. So the maturation place are different. The production place is the same. Okay, and then both of these mature B cell and T cell then will go to the special organ like the lymph nodes. This is the place where this mature B cell and T cell stay. So it's just like if they are army, so this lymph node is just like the place for army to stay. Okay, so B cells and T cell productions, again, both of them are produced in red bone marrow. The B cell maturation is present in the red bone marrow, so they stay there in the red bone marrow, while the T cell maturation takes place in the thymus gland. And this is the place for T cell maturation. Now, what is our enemy? So the enemies that enter into our system is called the antigens. The antigens are anything that enter into our body that might cause disease. Okay? It may be bacteria, okay? So bacteria, viruses, okay? Bacteria, viruses, maybe fungi. They are type of antigen. Or maybe for renal cell. Like for example, if we have blood transfusion, that the cell that enter into our system is become antigen, become our antigen. Or maybe part of the plant molecules, okay, like pollen, for example. That's allergen. Allergen is also considered as the antigen. Or maybe hair, dandruff, animal hair, part of skin of animals, or maybe some other things that enter into our body that have the potential to cause disease, they all call as the antigens. Okay, so antigen is anything that enter into our body that provoke our immune system. Okay, so there are some classification of the antigen. For example, alloantigens, and this is cell surface marker of one individual that are antigen to another that the same species. So like for example, blood transfusion, we receive cell from other people. That type of antigen is called the alloantigen. It's coming from the same species. Super antigen is the antigen that will provoke our immune system, maybe coming from bacteria, viruses. Allergens, this is special antigens that provoke the basophil and the IgE, right? So remember, basophil is the one that fight against allergy, and the antibody that also help the basophil is called the IgE. So this is allergens. Autoantigens, this is our own cell that actually provoking our immune system. And usually, if it is happening, we have a disease, what we call the autoimmune disease. Like for example, RA, rheumatoid arthritis. This is a type of autoimmune disease when our antibodies is actually attack our own cell, especially in the joint area. Why? Because the own cell is actually produce the markers that our own antibody recognize this as a foreigner, even though it's actually our own cell. So that's what we call the autoimmune disease. This is just an example. There are some proteins, but when it is present by itself, it will not provoke the antibody. So this is not antigen. Remember, antigen is the one that provoke our immune system. However, if the small proteins bind to something, another protein, for example, then it's become bigger, then it will provoke the immune system. Then this one is become antigens. So antigens is anything that will provoke our immune system. Now, what's immune system that usually provoked by the antigens? They are called the antibodies. So antibodies will always try to kill or destroy antigens. Okay, so we'll always do that. So antibodies kill the or destroy the antigen. That's what we call the reaction. So sometimes we talk about the antibody antigens reaction, because that's the normal reaction of antibody, which is try to destroy the antigen. And just remember, we have 5 types of antibody. The most abundant one is IgG, IgA, IgM, IgD, and IgE. So every time we have the antigens enter into our body, then this into our body, then this antibody will be produced. uh, so they are produced. Okay, they are produced whenever there is an antigen present. So these antibodies then will try to kill these antigens. So that's the process. So the antibody is not always present in our system. It is only made whenever we have antigens enter into our system. Again, this is the 5 class of antibody, the IgG. The monomer is only one type of structure, one side. The IgA, it is dimer, okay, 2 sides. The IgM is actually 1, 2, 3, 4, 5. So there are 5 sides of reaction over here. So therefore it's called a pentamer. So IgM pentamer. The IgD is also dimer. Sorry, monomer, only one side. Okay, so this is one side over here. And the IgE also monomer. Okay. So IgG is the most abundant one, 80% in our blood. IgA about 20% or 30% in the liquid, okay, like mostly in saliva, sweat, tear, for example. IgM about maybe 6% in the blood, IgD about 1%, and IgE less than 1% in the blood. Right? This is the one that we talk about, that antibodies will always try to destroy the antigen. That's the process. So antibody is our weapons, the antigen is our enemy. So always the weapon will always try to kill the enemy. So this is what we call the antibody antigen reaction. So how? Now the question is how the antibody destroy the antigen. There are several ways. The first one is called the opsonization, which the antibody is actually coat the antigen, like the microorganism, and make them become inactive. It's not really destroy this antigen, but make the antigens inactive. It will not cause disease. Agglutination, this is when the antibody is actually bind to the whole antigen and then break the antigen and make them combine together, make a bigger molecules that will not be active. Get a large clumps of antibodies antigen reaction. And the last one is what we call the neutralization, where the antibody will bind to the antigen and then the antigen become inactive, become neutral. It will not be able to cause disease. So this is the some process of antigen antibody reaction. Okay. Again, this is the maybe the picture of that process. Like this one, for example, opsonization, when the antibody bind to the antigen and then make the antigens become available for the phagocyte. In this case, it's macrophages. So when these uh, antigens over here, which is bacteria, bind with this antibody, is become available for the macrophages to eat them. Okay, so this is what we call the opsonization. Neutralization, like for example, this antibody bind to the antigens like virus over here, and then causing the virus become neutral. You see the spike and everything will be covered by the antibody. Therefore, it will not be able to causing infection. This is the one that is normal virus over here, which is where it will be able to cause infection. But whenever this virus is covered by the antibody, it's become neutral. This is what we call the neutralization. Agglutination is when several antigens combined together with this antibody is become big clumps of molecule, and this big clump of molecule then will be precipitated, will not causing the infection. Okay, so it become inactive. Complement system, we still remember the complement system where the protein actually dig the holes in the bacterial cell. The same thing, the antibody is also able to produce similar action, which is digging the holes of the bacterial cell. This is the same name, it's become complement fixation. The other one is by combining these viruses with many antibodies. This is antibodies antigen reaction. So the virus is antigen, and then it's become precipitated. When it is precipitated, then it will not cause the infection or disease. So this is how the weapons, which is the antibodies, kill or destroy the antigens or microbes. Okay. The primary response, this is really important when we talk about COVID-19, maybe you've heard about this one, that the one that will increase and the antibody that will increase will be the IgM. Okay? So the IgM will be increased first. That's the primary response. This is when somebody have a positive COVID-19 positive, for example, they're actually measuring the IgM antibody. After several days, then it's produced the secondary response. In this case, then the one that is produced a lot will be the IgG. Okay, so the IgG will be increased as the second response, and this IgG is actually the one that will destroy the antigens, like the COVID-19, for example. So when our IgG increase, that's a good sign. That's a good sign because that's the time when the antibody, in this case IgG, will destroy the uh, antigens. Okay, again, this is the first response. The IgM will be increased first. This is, then we have the positive indication, and then the IgG will be produced later. And then in the second response, the IgG will keep steady produce. IgM actually will be reduced in production because they are not needed anymore. Mostly the IgG, the one that will fight these viruses. This IgG over here is the one that will fight against the viruses. So the first response usually take about 2 to 5 days, and the second response, let's say it is COVID-19, about 2 to 5 days during incubation period. And then this one, the destruction of the viruses maybe take about 7 to 10 days. And this is when the IgG destroying the viruses. That's the time when we have the disease, the illness. Now for the specific immunity, again, this is the third line of defense. They are specially done by the special army, which is the B cell and the T cell. Okay? So there are several types of T cell actually. The first one is called the T helper, TH. Okay? Sometimes it's also known as a CD4. Okay? So they are developed from CD4 into T helper. The second one is called the cytotoxic T cell, or sometimes it's called TC. And they actually develop from CD8. And so when we talk about CD8, this is the same as the TC, cytotoxic T cell. Another type of T cell is called the natural killer. This is the T cell that lost their specificity, so they are actually able to fight any type of infection. But this T helper and TC are the one that is really important for our immune system for specific immunity. So the TC, when they destroy the infection, is actually by destroying the infected cell. So destroy the infected cell. So therefore, this type of immunity is called the CMI, cell-mediated immunity. So the TC actually destroy the cell. This is how they perform the action. Okay, so this is the TC, the cytotoxic T cell. When they have, let's say our cell have the infection over here, then the TC will produce the toxin and enzyme. This toxin and enzyme then will enter into the infected cell and kill the infected cell. So this is what we call the CMI, cell-mediated immunity. So that TC is actually producing the toxin and enzyme that will destroy the infected cell, and the virus will be also destroyed. Some of them then will be available on the interstitial fluid, and this has become the job of the antibodies to kill them later when it is outside the cell, because the antibodies is actually unable to enter into the cell. So antibodies like IgG, they only destroy the antigens that's located outside the cell. The one that is inside the cell will be destroyed by the TC. Okay. Now, this is just a summary examples of how the immune system works. Examples, this is normal, during normal healthy cell condition. Okay, remember the TC will always survey. This is like a sheriff that always survey our body. If there is nothing happen, then say, okay, you okay. There is another cell that also perform surveillance. This is actually the T helper also perform surveillance. So say we can call this as a deputy sheriff. Okay, so the T helper also survey, but T helper only survey the cell that able to eat bacteria, which is like macrophages, for example. The TC will survey other cell, all cell, except the macrophages and phagocyte and neutrophil. So all cell will be surveyed by TC, macrophages or phagocyte will be surveyed by T helper. So this is normal condition, there is nothing, so there will be okay. So it's just survey and then check, just like sheriff and deputy sheriff survey the community, and there is nothing happen, then they say, okay, good night. Have a good night, and they say goodbye. Okay? But if there is something happen, like for example, chickenpox viruses enter into our system, then what happen? It will causing infection into this cell. So this is infected cell. So when the cell get infected, then it gives the marker, okay, give the sign. So this is the time when the TC will know that, oh, okay, there is an infection, then the TC will become active. When it is active, it will make other TC, it's called the TC clone, and also it will make the TC memory cell. Okay, now when the chickenpox enter into our system, the macrophages also know, okay, there is an infection. So this macrophages try to eat this infection, and it will be recognized by the T helper. Okay, remember T helper is the one that survey the macrophages. And whenever the T helper have that signal, then it will develop into T helper clone, which is active T helper, and T helper memory cell over here. Yeah. Right now we forget about the TC memory and T helper memory because it will work later. The one that work right now is the T helper, the active T helper over here. It will produce the chemical, especially it's called the interleukin. The interleukin will go to the TC, give the activation to the TC, and also give the activation to the B cell. So this is the B cell coming over here. When the B cell get the activation from the T helper, and then it will become plasma B cell and B cell memory. Okay. The plasma B cell, then the one that produces the antibody. Remember, antibody produced by the B cell, which is plasma B cell. These antibodies then will destroy the chickenpox viruses that is outside the cell. The one that enter into the cell then will be destroyed by the TC by producing the toxin and enzyme. It will destroy this infected cell, and then the virus will be available for the antibodies to be destroyed. Boom. This process happenings about 7 to 10 days. This is the time when people with the chickenpox infection will have the disease, about 7 to 10 days. The same thing with the flu, okay? Maybe 7 to 10 days. After that, the virus will be gone, and then we become better. We cure from the disease. Then the antibodies also will be destroyed, will be gone, okay? Because they are made, they will not be available forever. They also will be gone. So after 7 days, 10 days, we have become better. We are, become healthy again. We cure from the disease. Now, when the second chickenpox virus infection coming, after several months, for example, then we will not perform this process again. Because what? Because we have this memory cell. The memory cell will recognize it, that chickenpox, and then they will destroy it right away. So we don't have the disease anymore. This is what we call the immunity. So we become immune against chickenpox after we have the infection. But if the one that coming, the next one is coming is polio virus, for example, then we have to perform this process again. So we have to perform this process again, special for polio viruses, and etc. For other viruses, then they will be have the similar process like this one. So this is how our body exactly protect us against the specific infection. After the infection, we fight them and we become cured from the disease, and at the end we become immune to that specific disease. Okay, so that's how the T cell and B cell works in our system. Now, right now we have what we call the vaccination. So vaccination is almost similar with the infections of the disease, but this vaccination is considered as a weak pathogens. Okay, it will cause our system to produce memory cell without having the illness. Okay, so this is what we call the artificial active immunity. So after we have this vaccination, then we become immune to that disease. Okay, like for example, chickenpox vaccination. So the kid that have chickenpox vaccination will not have the chickenpox disease, but their immune system able to produce their memory cell that will protect them against the real chickenpox viruses. Okay, so the vaccination is actually the weak viruses that is injected into our body. in order to produce the memory cell. Now, vaccines can be prepared, maybe the whole cell that being killed or become weaker. Live cell that is weaker. Maybe part of the antigenic molecules that coming from the cell, bacterial cell viruses, or maybe using the genetically engineered microbe to produce the vaccine, or maybe part of the toxin, the exotoxin that produced by bacteria, converted into toxoid. These are the type of vaccine that is usually used for vaccination. So this is the picture. So the whole cell over here. So the whole virus is actually injected, but that virus that injected is weaker. It's a weaker virus. Or maybe just part of, of the pathogen. Like for example, maybe just part of the spike of the viruses, part of the cilia or whatever part of the cell wall, for example, of the bacteria injected into the cell. So of course, because this is only part, like just a spike injected into the cell, then it will not cause disease, because the one that causing disease is actually this one over here, the DNA. So if only injected the spike, it might increase our production of memory cell without giving us the disease. Or maybe using the recombinations, using bacteria to produce special vaccine. So this is how the vaccines is actually being made. This some requirement for effective vaccine. It has to be low levels with low adverse side effects or toxicity. Should protect against the exposure of natural pathogens. Should stimulate the B cell and T cell responses, which is creating the memory cells. And should have a long-term, long-lasting effects, and should not require higher dose or maybe booster. That's the better vaccine. Should be in infancy. It's not expensive and can be easy to administer. Maybe oral vaccine or injected vaccine. Administration mostly injections. That's the most common type of vaccination. Some vaccine also use the oral administration and maybe nasal administration. Now now we're gonna talk about the immunity. So immunity is the condition when someone is actually immune against specific disease. Okay, there are several type classification, which is active immunity. Okay, this is when the person actually have or challenged by the antigens directly. It will stimulate the memory cell and creating the immunity. And usually, if it is active, it's become long-lasting. Passive immunity, this is actually when the person receives the antibodies. There is no production of memory cell, so just receiving the antibodies. Therefore, this becomes short term because antibody is not forever, right? Maybe 3 months, 6 months. Natural, if it is the challenge is actually part of the natural disease, like for chickenpox disease, that's become natural. Artificial, if it is given through maybe injection, like vaccine, for example, that's become artificial. Now, if we combine them together, then there are several types of immunity. The first one is called the natural active immunity. And this is natural, is mean from the natural cause, for example, chickenpox disease. So after chickenpox disease, and then what happened? We will produce the memory cell. It's become active immunity. So therefore, natural active immunity will be long-term immunity. So this is after getting the chickenpox disease, we produce the memory cell, and then we become immune for a long time, okay, against the chickenpox, for example. The natural passive immunity, remember passive mean antibody. So this is actually someone that receive antibodies naturally, okay, from natural cause. For example, children, baby that receive antibody from his mother during the embryonic stage through the placenta or during the breastfeeding. Because breast milk is actually have antibodies on that, and this becomes short-term immunity because antibody is not forever. So it's short-term, but it is given through the natural cause. Artificial active immunity. So artificial means injection. So it's going to be acquired through injection or inoculation, like vaccination, for example, or flu shot. This is artificial. It's become active because the one that is injected is actually part of the microorganism. So our body will create the memory cell. It's become long-term immunity. Okay, so chickenpox immunization or vaccination will create the long-term immunity, just the same like as coming from natural active immunity. Here, artificial passive immunity, this is through injection of antibodies. Okay, injection of antibody. For example, if nurse taking care of hepatitis B patient, usually he will receive the injection of hepatitis B immunoglobulin to protect him against the hepatitis B disease. And again, it's going to be short term because antibodies are short term, right? About maybe 3 to 6 months. Okay, this is the picture of that type of immunity. If it is natural, okay, maybe active, like having the disease, or passive, which is the kids receive the antibodies from their mother. If it is active, it's going to be long term. If it is passive, it's going to be short term. Okay. The artificial is injection. This one is vaccination, which is injection of the disease, weaker disease. Then it's become active, it's going to be long term. Passive is just like this kid receive antibodies, injection of antibodies, it's become passive. And this passive is going to be short. So short-term immunity for passive. One more is about herd immunity. So when many people get vaccination, then the whole community is become protected. Even the one that do not get vaccination will also get protected because most people is actually immune against the disease. So this is what we call the herd immunity. So the herd immunity, results from uh, mostly vaccination. When most people get vaccinated, then the whole community will be protected against that disease. Okay, that's what we call the herd immunity. I think that's all for chapter 14 and 15. We'll see you again with the next chapter, which is chapter 16, disorders or abnormality in the immune system. Goodbye. Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 16, which is disorders in immunity. So on chapter 14 and 15, we talk the normal action of our immune system. Now we will discuss some disorders in our immune system. Okay, it can be divided into two types. The first one is underreaction. So underreaction means our immunity or our immune system is actually not working properly. Maybe first the T cell, okay, or the immune system do not survey our body. Okay. Second, maybe it is caused by loss of immunity, like for example, deficiency of T cell or B cell, or maybe both of these T and B cells. That's under reaction. The next one is actually overreaction. It means that our immune system actually overreactive to foreign molecules. Yeah. Normally, that foreign molecule usually do not cause disease to us, but our immune system try to destroy them. This is what we call the overreaction, or sometimes it is also called as the hypersensitivity. There are four types of hypersensitivity. The first one is called the type 1 hypersensitivity, and it is caused by the uh, antibodies, especially the IgE, that is overreactive. So that's type 1. The second one is called the type 2 hypersensitivity. It is also caused by antibody that is overreactive. In this case, the IgM that is really overreactive for type 2 hypersensitivity. The third one is called the type 3 hypersensitivity. It is also caused by the antibodies, mostly the IgG that is overreactive. And the last one, type 4, this is the only one that is caused by the cell, which is the T cell that is overreactive. Okay, just remember type 1, 2, and 3, it is caused by the antibodies, okay, or the Ig. The type 4, this is the one that is called by cell, which is the T cell that is overreactive. Okay, we start, okay, uh, again, this is just a picture disorders in immunity. Again, it is divided into two conditions. The first one is underreaction. Okay, so maybe caused by lack of surveillance. And if this happens, usually it will cause cancer. So cancer is usually caused by the lack of surveillance of our immune system. The second one is the loss of the immunity, maybe loss of T cell or loss of the B cell. Okay, so our body do not produce T cell or B cell, or maybe loss of both B and T cell. And this is what we call the immunodeficiency. So immunodeficiency disease caused by lack of our immune system. And the second type of these orders in immune system is the overreaction. If it is caused by the B cell, remember B cell, they produce antibodies, okay, or IG, then it's going to be type 1, okay, type 1, which is caused by the overreactivity of IgE, antibody E. Type 2, this is also caused by the antibody, especially antibody M, okay, IgM. And type 3, this is also caused by the antibody, mostly the IgG, that is overreactive. So this is the first type of overreaction that is caused by the hypersensitivity or overreaction of our antibodies. The second type, it is caused by overreaction of our T cell. Okay, so the T cell over here, and this is what we call as a type 4 hypersensitivity. Okay, we start with underreaction. Remember, there are two types. The first one is lack of surveillance. You should remember that our immune system, they usually survey our body. If there is something happen, then they will try to destroy that foreign cell. And the surveillance usually done by TC and the T helper. Okay, so when the TC survey our body, if they recognize something happen, like maybe the cell that reproduce or divided uncontrollably, like a cancer cell, then the TC will destroy the cancer cell. However, if the TC is actually lazy, okay, which is do not really perform the function to survey our body, then the cancer will develop. Okay, so the cancer mostly develop because of our T cell, especially the TC that do not perform the surveillance. Okay, so just remember this process. So how the cancer actually occurs. This is normal uh, condition over here where the TC, okay, they survey the cell. So let's say this is our cell, and then the TC will have the receptor to recognize the marker over here. Okay, so the marker over here. So when there is nothing happen, then the TC will say, okay, you don't have a problem. But if there is something happen, like for example, cancer cell develop over here, okay, then the TC recognize this problem, it will produce the toxin, and this toxin will destroy this cancer cell. Okay, so that the normal action in our immune system, where the TC actually always survey our cell, our system. If there is cancer cell, it will destroy that cancer cell. So just for your information, all cells in our body have the capability to be cancer cell. And the reason we do not have cancer because of this TC that always perform its function to survey and also to kill this cancer cell. Now, what happens if the TC is actually not active? Okay, so lack of surveillance. So the TC is here, and then the cancer cell develop over here. So the cancer will grow because the TC will not kill this cancer cell because of lack of this surveillance. So this is how the cancer develops. So this is one of disorders in immune system. So some people maybe have the TC that do not really perform the surveillance and destruction of cancer cell, and they will develop the cancer. The second type of underreaction, it is loss of immunity. And this is also known as immunodeficiency disease. There are two categories of this immunodeficiency. If it is happening since uh, embryonic development, this is called the primary immunodeficiency. So usually that person will be born with this deficiency. So he actually unable to protect himself against disease because he does not have the immune system, okay, the part of the immune system. The second category is called the secondary immunodeficiency disease, which is the disease is actually acquired after birth. Okay, so when he was born, he actually have a normal immune system. But after that, there is something happen, for example, maybe infection of HIV. Okay, when this person has infection of HIV, then this HIV actually will kill or destroy many T helper. And because this person do not have T helper, then he will develop the immunodeficiency, which is AIDS. Okay, so AIDS is the famous example of secondary immunodeficiency disease. Okay. We start with the primary immunodeficiency disease, which is someone that born with this condition. He does not have maybe B cell or T cell or maybe both. So he doesn't have both B cells and T cell. And if he doesn't have both T cell and B cell, it is called a severe combined immunodeficiency or SCID. So he actually lack of both B cell and T cell. Example of the one that only lack of B cell is agammaglobulinemia, and usually these patients will unable to protect himself against mostly bacterial infection. So he must have a lot of bacterial infections. The one that do not have T cell, okay, or T cell defects, okay, they usually or he usually have a lot of viral infection, okay, and also parasite. Okay, so this is the difference between B cell defects and T cell defects. For the one that do not have both, then it is going to be severe, which is unable to protect himself against infectious disease. Basically, this person will mostly die because of infection during early ages. As this is examples of this immunodeficiency. This is primary immunodeficiency disease. Okay, during the embryonic development, this person maybe do not produce the T cell, but the T cell is actually do not develop into the thymus gland. And if it is happening, then this person has a disease called the DeGeorge syndrome. If this person's able to produce T cell, and the T cell is actually developed in the thymus gland into mature T cell, but unable to perform the function, then this person has the disease called the ADA. And again, mostly if they do not have the T cell or lack of the T cell activity, he will have a lot of viral infection, parasite like fungal infection, protozoa infection. Okay, so this the one that do not have T cell or lack of the T cell activity. Now, how about the one that do not have a B cell? So if it is happening very early, then it is called a congenital gamma globulinemia. Okay? If it is happening after development into the mature B cell, okay, then the condition is called the hypogammaglobulinemia. And again, usually, if he does not have the B cell activity, he will have a lot of bacterial infection during his life. So the infection will be happening a lot. So recurrent bacterial infection. Now, how about okay? So let's see again these tables. The name of the disease, if it is B cells deficiency, there are several disease, and there are several primary immunodeficiency that is caused by the B cell defects, a gamma globulinemia, hypogammaglobulinemia, some selective immunoglobulin deficiency. That if it is the lack of B cell action or activity, if it is lack of the T cell activity or T cell defect, maybe thymic aplasia or chronic mucocutaneous candidiasis. This is called also the George syndrome of the first one. If it is both B cell and the T cell defects, then the disease called the SCID, severe combined immunodeficiency disease. Example is the ADA deficiency, XSCID deficiency, ataxia syndrome. This is examples of boy that have this SCID. His name is, I think it's David, the bubble boy. So when he was born, doctor actually know that he has this SCID, and doctor put him in kind of place that is protected from infection, like in the aquarium, something like that. Okay, in the place that covered with the plastic, and and the doctor try to learn, try to protect him from infection because he actually did not have the ability to fight any type of infection. So doctor actually able to keep him for 8 years. But after 8 years, he actually finally passed away due to the infection. You can imagine, doctor already tried to protect him in a very secure, sterile place, but still he actually got the infection. He passed away because of this infection. So this is a very severe condition, and usually if it is not helped, uh, the patient will die soon after he get born. Okay, the secondary immunodeficiency, this is the immunodeficiency that acquired after birth. And the famous example is AIDS. Okay, So AIDS is actually destroy the T helper, and then the people with AIDS, they usually die because of the infection, not because of the HIV itself, but because of just regular infection, maybe diarrhea, pneumonia, influenza. It can cause, it can kill the AIDS patient. Another example is leprosy, tuberculosis, measles, and other disease like the one that may be causing cancer and diabetes. And maybe due to the nutrition deficiency, stress, pregnancy, and aging, that also can causing the immunodeficiency. But the most severe one is the AIDS, okay, acquired immunodeficiency disease. This is due to infection of HIV viruses. Okay, again, how AIDS develop. Okay, remember when the HIV virus enter into the cell, it has special receptors on the T helper. So T helper over here has special receptor for the HIV. So the HIV virus will attach and enter into T helper and destroy many T helper, until it's very low concentration. Then that person will develop the AIDS. When the T helper destroyed, then the T helper will not be able to activate the T and the TC. The T helper over here will not able to activate the B cell, to produce the antibodies over here. Okay, so people with AIDS, then it will not be able to destroy the virus and kill the virus by producing these antibodies. So basically, patient AIDS, AIDS patient will not be able to protect himself against many infection, many disease. And again, mostly they will die because of the regular infection. Okay, we finish with the underreaction. Now we discuss the second type of disorders in immune system, which is overreaction or hypersensitivity. Again, there are four types of hypersensitivity. And the first one is called the type 1 hypersensitivity. And this is mostly known as allergy. So allergy is not normal. This is actually abnormality. This is a disorder in immune system. Again, the allergy usually caused by the IgE that is hyperreactive. Okay. The second one, it is called the antibody mediated hypersensitivity, and this is mostly caused by the IgM, right, that is hyperreactive. Example is the blood group incompatibility, which is if someone with type A blood give transfusion to type B blood, then the blood will not match. The antibodies of the receiver, the recipient will actually try to destroy the red blood cell of the donor. Okay, so the blood that enters the human body is actually and not something that is dangerous. Okay, if it is sterile blood. So, but the IgM try to destroy this blood and causing problem to the recipient. So this is the type 2 hypersensitivity. The next one is type 3 hypersensitivity, is also known as immune complex mediated hypersensitivity. This is mostly caused by the IgG that is hypersensitive. And this one mostly causing disease, what we call the autoimmune disease. So many autoimmune disease is actually in type 3 hypersensitivity, which is the antibodies IgG that is really, really reactive and try to destroy any cell. Sometimes that cell is our own cell, therefore it's called immune disease. And the last one is type 4 hypersensitivity or T cell mediated hypersensitivity. And this is the only one that caused by the cell, which is the T cell. Remember the other one over here, the other three is caused by the antibodies, IgE, IgM, IgG. And this one, the only one that caused by the T cells. Okay, we start with the type 1 hypersensitivity, okay, which is allergies. And this is caused by the hypersensitivity of IgE. Okay, so the IgE or antibody E that is really, really hyperreactive. There are two classes of allergy. The first one is called the atopy, which is local allergy, and anaphylaxis. This is a systemic allergy, which is goes to the blood circulation. Okay, so just remember that allergens, this is mostly harmless. This is not pathogenic microorganism. So this is mostly harmless. So for normal people, these allergens will not cause any disease. So for normal people, they actually do not try to destroy these allergens. But for some people with the hypersensitivity, the IgE really, really hyperreactive is try to destroy allergen. Okay, and when they try to destroy allergen, it will produce what we call the allergy mediators, like histamine, for example. And this histamine actually will produce what we call the allergic reaction. So some people that have allergy, they usually produce the allergy mediators like histamine, bradykinin, and then finally he will develop this allergic reaction, like for example, runny nose, itchy eyes, and etc. Okay, so the allergens, maybe we will see how they actually enter into our system. Okay, so the major allergens and their portal of entry. There are many allergens actually that can cause allergic reaction or allergy. If it is go through the respiratory tract or inhalants, is like pollen, dust, mold spore, danders, animal hair, formalin aerosol. They usually use the respiratory tract. Therefore, they are called as inhalants because we inhale that allergen. If it is go through food or drink, it's called the ingestion. Maybe food like peanut, shellfish, soybean. Okay, some people may be allergic to eggs, for example. Okay, food additive, drugs like maybe oral drug, aspirin, penicillin, for example. Uh, if it is injected, like a bee sting, for example, it is called the injectant. So bee sting, insect bite, drugs, injected drug, like vaccination maybe, okay, serum enzyme hormone, injected hormone. That is called as the injectants. And the one that attach or contact to skin, for example, they are called as the contactants, like maybe topical drug, the antibiotics that we put on the skin, cosmetic, heavy metals, detergent, formalin, rubber. Some people have allergy to rubber, solvent, nice. So this is a classification of allergens based on the portal of entry, which is inhalants, ingestants, injectants, and contactants. Now, the mechanism of allergy, it's like developed into two stages. The first stage is called the sensitizing dose. This is when the patient or the person have the first contact with the allergen. And it will develop the dose that will go to the provocative dose, which is the development of the allergic reaction. So the allergic reaction is happening in this provocative dose. And sensitizing dose usually no sign or symptom of allergy. Okay, it's just sensitizing. This is the first development of allergy. And usually this sometimes take maybe days, weeks, okay, months, some people maybe years after that contact, they develop the uh, allergic reaction, which is the second stages over here. But for some people, maybe after several days or getting the first dose, then right away they develop the allergic reaction, which is the provocative dose. So okay, so this is no wonder that some people actually develop the allergy after several days, some people after several weeks, some people maybe after several months, even some people maybe years after the contact, he does not develop the allergy. Okay, it's really depend on the person, the condition of that person. The symptom of allergy usually rash, itching, redness, increased mucus discharge, runny nose, pain, swelling, difficult breathing, especially if he has asthma. So this is some symptom and sign of allergy. Now, we're going to see this process again, the stage. Okay? The first stage, which is the sensitizing dose. This is when the person receive the first contact with the allergen. Let's say this is pollen. Okay, so pollen enter into the respiratory tract, finally go to our system. Then the B cell, remember B cell, and the IgE is the one that uh, try to destroy the allergen. Okay, fight against allergen. So this the B cell have the receptor for these allergens like pollen. Okay? So when it receives the attack or attach this pollen, and what happened? The B cell, remember this B cell will uh, activate the plasma B cell, okay, to produce the IgE. So remember, IgE is antibodies, all the antibodies produced by the plasma B cell. And the one that produced due to the allergens reaction is called the IgE. So the IgE will be produced. Usually there is no sign and symptom in this time. After time, maybe again, maybe days, weeks, okay, maybe months or years, okay, depend on that person. So that IgE will bind to the mast cell, which is the basophil. Okay, so basophil is a type of white blood cell that produce the mediators. Okay. So this IgE will bind to the basophil. And again, if there are many basophil, will take maybe more time to produce this mediator. Okay, so when the basophil or mast cell produce the mediators, okay, so mediators, yeah, like histamine, for example, then this mediator will enter into the circulation. It will be carried into several organs like the eyes, causing the red eyes, watery eyes, nose, maybe runny nose, the skin, hive, rashes, itchy. So this mediator is actually the one that will give this symptom. And this is happening on the second stage, okay, which is the provocative dose over here. Okay, so that's the stages of allergy. Okay, just remember there are two stages. The first stage is called the sensitizing, which is mostly IgE production, and then provocative dose, which is the release of these mediators, okay, from the mast cell or from the basophil. So again, there are two things that is causing that allergy. The first one is the IgE, and the second one is the basophil over here. So remember when we talk about our immune system, the basophil, which is the white blood cell, and IgE, which is one type of antibody, they work together to destroy the allergens. Both of them is actually the least abundant type of immune system. So basophil is actually the least abundant type of white blood cell, and the IgE is actually the least abundant type of antibody. Okay. Again, this is the roles of mast cells and basophil in producing the mediator. It's called the allergy of chemical mediators. Like the most potent one is histamine. Okay, so this is the list of the mediators that produced by the mast cell and basophil. The most potent one is histamine. Then there is serotonin, leukotriene. This is usually for people with asthma. It's really dangerous for them, leukotriene, because it can constrict the respiratory muscle, especially on the bronchus and trachea. And then platelet activating factor, prostaglandins, and bradykinin. So these are the chemical that is produced by basophil and mast cell, and this is the one that will cause this sign and symptom of allergy, like rashes, itching, redness, rhinitis, sneezing, diarrhea, shedding tears, as a sign, some sign and symptom of the allergy. Now, if we uh, see one by one, like prostaglandin, this is one mediator. They usually dilate the blood vessel, okay, and here to the bronchioles, okay, also will actually constrict the bronchioles. Okay. It will go to the nerve cell and causing headache. So headache mostly caused by this prostaglandin. Other mediators like histamine, serotonin, bradykinin, yeah, go to the blood vessel, it's dilate the blood vessel. If it is go to the skin, for example, then the skin become red, okay, redness to the skin because the blood is actually going to this area. If it is go to the respiratory tract, the muscle, they actually constrict the muscle of the bronchioles, which is the smooth muscle over here. It's causing difficult to breathe. Okay? It's increase the peristalsis of intestine, causing diarrhea, vomiting. If it is going to the epithelial cell, it's causing the epithelial cell to produce more liquid, which is mucus. So you see runny nose, tear. This is caused by this mediators over here. Leukotriene, this is typically for the asthma patient, it can constrict the bronchioles. So the asthma people will have difficulty to breathe, sometimes causing death. Okay, so these are again the mediators that produce the allergic reaction. Prostaglandin usually causing headache, leukotriene disease for asthma people, it will constrict the bronchioles. For histamine, serotonin, bradykinin, this is usually the one that causing runny nose, watery eyes, difficult breathing, vomiting, and many more reactions actually. Okay, again, the most potent mediator is histamine. Okay? This is the one that usually have a very fast action, with this allergic reaction. Okay? This is the reason why many allergy medicine, okay, have antihistamine. So if you read, okay, the allergy medicines, mostly they have the antihistamines in their drugs, that medicine. Okay, there are several, again, two type of allergy. Yeah, if it is like high fever, it's called the atopic disease or local seasonal disease. Asthma, one of them. Eczema is another type of allergy. There is also food allergy. Okay? This is mostly caused by food like peanuts, for example. And drug allergy, this is caused by the medicine, drug, like antibiotic, for example. Okay. And if it is goes to the blood circulation, it is called the anaphylaxis. Example is bee sting, and sometimes it's become fatal to the person. Like a bee sting, injected antibiotics and serum injection. This is also can cause systemic anaphylaxis. So systemic anaphylaxis is considered more dangerous than atopic allergy. How to diagnose the allergy? Usually, if you go to the allergy clinic, they will test you, okay, using what we call the skin test, and will give you this chemical that's coming from different type of allergens, yeah, and will test you whether you have reaction or not. So this is positive reaction over there. This means that you allergy to this chemical that doctor give to you. So this one is negative, negative, negative. You can see the different on the skin if you have the allergy to the specific chemical that being tested on your skin. Okay. So this test is called the in FIFO test because it's actually the doctor injected something chemical inside into our body. So it passes actually by putting some chemical into our system. Treatment and prevention for allergy. The best one is actually avoiding allergy, which is very difficult. It means that you have to stay home. Okay, do not go outside during the allergy season, for example. So in spring, when there are so many pollens available, uh, then in order to prevent yourself against the pollen, you have to avoid that allergen. So stay home, okay, or maybe wearing face mask also will help protect against this allergy. Second is using the drugs that block the action of the mediators. Many drugs have what we call the antihistamine. So you take the allergy medicine, mostly they have antihistamine. For people with asthma, they also have to take the drug that have anti-leukotriene. Okay, so the drug usually have this ability to block the mediators like histamine or leukotriene or maybe bradykinin. And the last one is using what we call the desensitization therapy, which is injection of allergen or injection of antibody in order to block the IgE production. Usually, the IgG is the one that will be injected into the system in order to compete with the IgE. So that's the injection of antagonists, which is desensitization therapy. Okay, this is preventions against allergy, like for example, injection of corticosteroid, uh, taking drugs like antihistamine, like aspirin, epinephrine, anti-aspirin, and other type of drug in order to block the histamine or any other mediators. There is also some research showing that taking honey, especially water honey, will help to protect someone from allergic reaction that is caused by pollen. For the desensitization therapy, again, usually injecting a specific antibody like IgE, I mean IgG, and this IgG will uh, block the production of IgE, or the IgE actually will not bind to the allergen, because allergen will bind to the IgG. If it is bind to the IgG, then this IgG will not bind to the mast cell. So only IgE that will bind to the mast cell. Okay, if this IgE do not bind to the allergen, then there will be no production of mediators. Okay, so this is actually the principle of desensitization therapy, which is injection of IgG that will compete with the IgE. So if the IgE do not receive the allergen, then the mast cell will not produce the mediators like histamine. Of course, the desensitization therapy will not last long, because antibody injection usually stay in our body for about maybe 3 or 6 months only. So usually the person will have to take uh, several injections in order to protect himself against allergy. Okay, the second type of overreaction or hypersensitivity is called the type 2 hypersensitivity. Again, this is mostly by the IgM, okay, the overreactivity of the IgM. And this IgM, they mostly produce in order to protect ourselves from foreign cell. Okay, like for example, blood cell. So when we receive blood transfusion, we actually receive blood cell from someone, and it is considered as foreign cell. So the IgM, okay, the IgM will try to destroy this foreign cell. So this is the basic action of the IgM, and they usually try to destroy the foreign cell. So therefore, when we perform blood transfusion, we have to be sure that the IgM will not attack that foreign cell, which is we have to give the compatible blood type, because if the blood cell is compatible, it is not considered as a foreign cell by the IgM. So IgM will not uh, attack that foreign cell. Okay, so we have to uh, study a little bit about blood type. So blood type, there are two types of blood type, which is the ABO blood type. The second one is called the rhesus blood type. Next, so we start with the ABO blood type. So based on the ABO blood type, there is four blood types, which is A, B, AB, and O. And this is based on the antigens that is present on the red blood cell. Okay, So maybe we can go to this picture over here. So someone with the type A, so if your blood is type A, then it means that you have A antigens in the red blood cell. So this is red blood cell. Okay. If you are type B, it means you carry the B antigens like this, okay, on your red blood cell. So red blood cell carry this B antigens. If your blood is type AB, it means that you have both A and B antigen, okay, on the red blood cell. So red blood cell carry that antigen. If your type is O, then it means that you do not have the antigen A nor B on your red blood cell. So your blood cell do not carry these antigens. Now, if you are type A, again, you will have antigen A on your blood. Your plasma, okay, so the plasma the, will produce antibodies. So the antibodies that will be produced will be not the same as your antigen. Because what? Because the antibody, remember, always try to destroy the antigen. So antigen antibody reaction. That's the way our system protect ourselves against foreign cell, maybe disease, by producing the antibodies over here. Okay, producing antibodies. So these antibodies will attack and destroy the antigen. Now, if you have antigen A, of course, you will not produce antibody A. So you will produce antibody B. So you have antibody B. You produce the antibody B in the plasma. If your blood is B, it means you carry B antigen. Therefore, the antibodies that you produce will be anti-A. It will not B because B destroys B. If you have both antigen A and B, which is your type AB, then you will not produce the antibody A or B. So no antibody produced related to this blood. Okay. You still produce other antibodies, but you will not produce the antibodies related to blood, okay, antigen A and B. If your type is O, you do not have antigen, therefore you will produce both A and B antibody. Okay, so that's the normal condition in your body. If you are A, it means that you have antigen A, then you produce antibody B. If you are B, you carry antigen B in your blood cell, and you will produce antibody A. Right? If you are AB, it means you have both antigen A and B in your red blood cell, and your plasma will not produce antibodies, okay, related to this red blood cell. If your type is O, you do not have antigens in your on your red blood cell. Therefore, you will produce both antibody A and B. Okay, so you make sure you understand this condition. Now, again, summary this ABO blood type again. Remember, in your blood, you have red blood cell. This is the most abundant type of cell in your blood. Usually look like this. Okay, you have red blood cell. Okay, now if you are type A, then again you will have antigen A. So let's say round. Okay, so this is antigen A. That's simple. Okay. If you type your blood type is A, it means that your red blood cell carry antigen A. Now, if you are B, again, it's going to be simple. Your red blood cell carry the antigen B. That easy, right? So if your blood is B, it means that your red blood cell has the antigen B on it. If you are AB, it means that you have both antigen A, okay, and antigen B on your blood. Okay. Now, if you blood O, then no antigen. There is no antigens on your red blood cell. Okay. Now, if you are A, again, remember you have antigen A on your red blood cell, then you will not produce the antibodies A. You will produce the antibody that look like this, which is antibody B. This antibody B will be attacking to this antigen B. Okay, so the shape is the same. Okay, so if you are B, then you will not have antibody B, but you have antibody that look like this, round. Okay, the one that will attack this antigen A. So this one is called the antibody A. Okay, if you are AB, remember you have antigen B and also antigen A like this. Therefore, you will not produce this antibody because if you produce one of them, then your blood will be destroyed. So you will die. So it means that no antibodies. Okay. If you are O, look at this O, there is no antigen. Therefore, you will be able to produce the antibody A, the one that is look like round, okay, and antibody B. Okay, now this condition can be used to uh, understand the blood transfusion. Okay. Let's say blood transfusion is actually the donor give the blood, which is the red blood cell, to the recipient. Okay. Now, if you are A, okay, you should be able to give blood to other people that also A. So it is okay to give your blood to other people that also type A. Okay. Now, if you are A, okay, you try to give blood to B, you give it to B like this, boom, and then what happened? This antibody A that is present in people with a type B will destroy this blood. Okay, so basically you will not be able to give your blood to someone with type B blood. Okay, because what? Because the type B blood have the antibody A that will destroy the antigen A from the donor. Now, if you are A, can you give blood to the AB over here? Of course, yes. Because what? Because AB do not have antibodies. So it doesn't really matter if you give blood over here, there will be nothing that will attack or destroy this blood. So it means that you can give blood to, if you are A, you can give blood to AB. Okay. Now, if you are A, can you give blood to someone with O blood type over here? Of course not, because the A has antigen A over here, and this O have both antibodies. One of them, which is the A over here, will destroy the A blood. Therefore, you cannot give blood to O. So you only can give blood to A and AB. Okay. Now, how about if you are B? Can you give blood to A? Of course not, because B have antigen B, and A blood has antibodies B, so it doesn't match. Okay. Can you give blood to yourself or someone with a B? Yes. So B can give blood to B. Can you give blood to AB? Yes. Remember, AB do not have the antibodies. Then AB can receive that blood. No problem. Can you give blood to O? Of course not, because O have this antibody B that will destroy this antigen B. Okay, so you cannot give blood to O if you are B. Now, how about if you are AB? Okay. So AB, remember, has both antigen over here. Antigen B, antigen A. Can you give it blood to the A? No, because A have the antibody B that will destroy this antigen B. So you cannot give blood to the A people. Now, but can you give blood to B? No, because what? You have antigen A over here, and the B have antibody A. This does not match. Okay? Can you give blood to the AB? Yes, of course. You can give blood to AB person. Now, can you give blood to O over here? Of course not, because O have both antibody A and antibody B that will destroy the antigen B and also the antigen A. So you cannot give blood to O. So basically, AB can donor to other AB people only. Okay. Now, how about if you are O? Okay. Remember, if you are O, you do not have antigen. No antigen. It's very smooth like this. Okay. So can you give blood to A? Remember, A have antibody B. It might destroy your blood. No, because what? Because you do not have any antigen. No antigen. So it doesn't matter. Yeah, you give it to A with the antibody B over here, then he still be able to receive it. So you can give blood to A. Now, can you give blood to B? B have antibody A. You don't have antigen, so it should be okay. So you can give blood to B. Can you give blood to the AB? Yes, both. No problem. No antibodies, no antigen. It's very, very safe. Okay. So yes, you can give blood to A. Okay. Now, can you give blood to O? Of course, O can give blood to O, even though O have antibody A and antibody B, but there is no antigen to be attacked. Therefore, it is safe. So you can give blood to O. Okay, now you can see over here that O can give blood to any type of blood, any blood type. Therefore, O is also known as universal donor, because someone with the blood type O, he can give blood to any people with any type of blood type. Now, if you see over here, you can give it to AB, AB can give to AB, AB, B also can give to AB, and A can give to AB. So it means that AB can receive blood from any blood type. It is the reason why the AB is called universal recipient. Okay, so someone that can receive any type of blood. So if your blood type is AB, it means that you can receive any blood type. If your blood is B, you are universal donor. Okay, so O is really generous. While AB is only give to AB only, but he can receive to from any blood type. Okay, so that's the basic uh, informations about the blood type and also blood transfusion. Why A cannot give to B, B cannot give A, okay? Why AB can receive any blood. Okay? So you know this theory. Why O can give anybody, because O does not have antigen over here, so safe for him to give it to everybody. And then AB does not have antibody, so therefore he is safe to receive any blood type. Okay, this is information about the donor A, for example. Remember, if the recipient is B, it has the antibody A. This is antigen A. And A and A will destroy. Okay, so that this antibody actually will destroy this antigen. And what happens? That blood will be clumped in the people that have get the blood transfusion, and finally it might clog the circulation. It can kill the uh, recipient. Another, the second type of blood type is called the rhesus blood type. Okay, so let's see. I'm gonna explain again how it look like. Okay, rhesus blood type. So the same. So if it is positive, he has the red blood cell. Let's look like this, red blood cell. Okay. If he is positive, rhesus positive, then it will carry antigens like this. Okay, let's say this is called the antigens RH. Okay. Now, the one that negative is mean that he does not have antigen. No antigens RH. Okay. Now, because this person has antigen RH, then there will be no antibody RH. Okay, because he has antigen over here. So the one that negative, he or she actually will produce antibody RH. It's gonna be look like this. Okay, so this one will attack the antigen RH. Okay, now transfusion, of course, the positive can give to the positive. They are the same blood type. So can positive give it to negative? No, because negative has antibody RH over here that will destroy this blood. Okay. Now, how about if negative can give it to the positive? Yes, because what? Because remember, negative do not have antigen. So it is safe to give it to positive. And positive also do not have antibody. It's really, really safe. Yes, the negative can give to positive. But negative can give to another negative? Yes, of course. Okay. So from this information, then we know that negative is also universal donor. Okay. The positive will be universal recipient. Okay, because it's positive, positive receive. and this negative give, etc. Now for this information and also from the ABO type information, then you can give the conclusion that the AB, okay, AB negative, I'm sorry, we talk about the O negative is the real universal donor. So if your blood type is O negative, you can give it to anybody. It doesn't really matter. Okay. If your blood type is AB positive, this is the real universal recipient. Okay, you can receive blood from anybody because your blood is AB positive. So your AB will be safe against the ABO type, and the positive will be safe for the RH type. Eh? O negative, the O will be safe for giving to anybody in the ABO type, and the negative will be safe to give anybody with rhesus blood type. Okay, now we can relate that information to condition. It's called the hemolytic disease of the newborn. And again, this is based actually on the rhesus blood type. Remember the previous PowerPoint over here, okay, someone with negative over here, okay, he will or she will produce antibody rhesus, okay, like this. Because what? Because his blood or her blood do not have the antigen, okay. The one that positive, it will has antigens, rhesus, okay, but no antibody. Okay, no antibody rhesus. Okay, just remember this. So if it is woman, if it is woman, it means that she is able to produce the antibody RH. Okay, remember if it is woman. Now we can relate that information to this condition over here. So woman with the RH blood negative blood type, he able to produce this antibodies resist. If he married with man with the RH positive, so the baby mostly will carry the antigen RH in its blood. So what happens? Okay, the mother will produce the antibody RH and will kill this baby's blood. So the baby mostly will die because of this. And this is what we call the hemolytic disease of the newborn. So mother with negative blood type, it has to be careful if she has husband with the positive blood type because the baby will also carry the positive antigen and the mother will produce the antibody RH. It will destroy this and usually the baby will die because of this problem, because of the destruction of his blood cell by the mother antibodies over here. Okay. So usually women that have negative blood type will receive injection from the doctor, in order to protect her baby from this problem, from this disease. So maybe on the old time, there are some couple that always have miscarriages, miscarriages. This is maybe due to of this problem. So the mother or the wife actually has negative blood type, and the husband has positive blood type, rhesus positive blood type, and the baby usually mostly will be miscarriage, born, die uh, during birth, or even during the pregnancy. Okay, and this information can also, can be used for blood typing, which is typing your blood, knowing what is your blood type. So again, the blood A have the antigen A. If we put the antibodies A, then it will causing this problem over here. So the blood will be agglutinate or clump. Okay, so if it is compatible, then no problem. Okay, no clump or agglutination. Okay, so no agglutination. This is okay. So this is broken. Okay, broken blood. Okay, so it can be used for uh, identifying your blood type. Let's say usually we put 3 drops of blood on maybe on the disc. Okay, so this is drop number 1, drop number 2, and drop number 3. On the drop number 1, we put the antibodies A. In drop number 2, we put antibodies B. And drop number 3, we put anti-rhesus. Okay, so we mix them. You see this one is no problem, no agglutination. This is no agglutination. What does that mean? So no agglutination mean this blood is actually O. It's telling you this blood is O because when you put the antibody A, there is no agglutination. It means that there is no antigen A. Right? When you put the antibodies B over here, no agglutination. It means that there is no antigen B in this blood. So the red vessel is just smooth like this. Right? So therefore it is O. Now when we put the anti-rhesus, antibody rhesus, and there is a clump, there is agglutination. It means that it's carry positive. Right? It's positive blood. So therefore the final identification, this blood will be O positive. Let's say we have another example over here. When you put the antigen A, antibody A, then this blood is clump. So it means that this blood has antigen A, because there is clump. When you put on the B, there is nothing happen. So it means that there is no antigen B. So if you have A and no B, it means that this blood is A. It's carry antigen A only. So therefore the blood is A. If I here, no, nothing happened. So it means that there is no antigens RH, which is called negative blood. Right? So at the end, the final identification will be that this blood is A negative. This is how to perform the blood typing. Okay. Now this is the table of compatibility for blood transfusion. Okay, so this is, remember, O negative. This is universal donor. Therefore, O can give it to anybody. So O can give it to O, give it to O plus. Okay, so all okay over here. So the O, which is the donor, can give to anybody. Now, and the recipient, then the B, the AB positive, is the one that can receive from anybody. So no problem. Right? But over here, And you have to understand that O positive cannot give to the negative because this positive and negative is not compatible. The O and O may be compatible, but positive negative is not compatible. A can give the O. So the negative may be compatible, but A is not compatible with O. A and O is not compatible, and positive and negative is not compatible, and etc. Okay, so this table maybe can help you to uh, perform the blood transfusion with uh, uh, safe way. Okay, this is the list of compatibility of blood transfusion. Okay, now the next one is the type 3 hypersensitivity. Again, this is mostly by IgG overreaction. Right? So we're gonna see the stages and the step of this hypersensitivity. First things, the antibody, which is the IgG. Okay, IgG will try to process the antigen. So this is antigens. Okay, the IgG antibody. Okay, so this IgG try to process these antigens. When they perform the process, they usually will bring that into specific organ, like for example the skin over here. Okay? So this become what we call the complex of antibodies, antigens, and also the organs. Okay, so complex. This is complex antibody antigen, and then it will lodge into specific organ like skin, for example. And then what happens when it is lodged? Then it will actually invite the neutrophil to come, and neutrophil, okay, will try to destroy this complex, but it's also destroying the organs. Okay, so it will cause the problem on the organ itself, okay, like the skin, in the kidney, joint, like RA, for example, and the heart, and things blood circulation. Okay, so that this neutrophil will try to destroy the complex and also will destroy actually the organ itself. The last one, which is the type 4 hypersensitivity. Again, this is the only one that caused by cell, which is the T cell. And if you want to see the process mostly like this, so like for example, the antigens enter into our system, let's say to the skin, it will be processed by the dendritic cell, and the T helper will come. And this T helper will activate the T cell over here, and the T cell will produce the toxin that actually try to kill or destroy this antigen, by destroying also the cell over here. And finally, what happens? There will be blister and inflammations on that place. Okay. Example is poison ivy. Is actually T cell mediated hypersensitivity. The T cell will also try to reject the other organs that is received during maybe transplantations, right? Organ transplantation, or maybe skin graft. So the T cell try to reject this foreigner cell or foreign cell. This is two principles. Okay, if the foreign cell is actually, like for example, the heart transplantation over here, then the T cell, the host T cell, will try to destroy the heart cell over here. Okay, because it's considered as foreign. Okay, it's called a rejection. Okay, if the one that the donor is actually the bone marrow, remember bone marrow produces the T cell and other blood cell. So if it is the organ that is transplanted is actually the bone marrow, then what happens? The T cell that produced by the donor bone marrow is actually the one that will destroy the host cell. Maybe this is the bone cell. Okay, so this is the principles of grafting or transplantation. So if it is organ other than bone marrow, the host T cell will try to reject that. But if it is organ bone marrow that is transplanted, then the T cell that produced by the donor bone marrow is the one that will destroy the host cell. Okay? Therefore, right, when performing transplantation or graft, we have to understand where the cell or the tissue coming from. If it is autograft, it means the cell is actually or the tissue coming from ourself, there will be no rejection. It's gonna be safe, okay? Because it's the same cell or the same tissue. Like for example, if someone have a problem with the skin on his or her face, it can be grafted from the skin from maybe lower part of her body. So there will be no rejection because the tissue is coming from itself or the cell is coming from itself. It's called a autograft. Another one that is safe, it will not be rejected, is called the isograft, which is the tissue coming from identical twin. Because identical twin, they actually have the same markers and the T cell will not reject that twin cell. So it will be no rejection. Okay. It is allograft. It is coming from the same species from human. Then the T cell will reject. Will reject the graft tissue. So there will be rejection. If it is coming from other individual, yes, different species, maybe from animal, again it will be rejection. T cell will reject that tissue. Okay, so therefore there is special drugs that usually doctor use during or after the transplantation. It's called the immunorepressive drug, especially if it is graft that coming from other people and maybe from animal organs. Okay, so now what we're gonna discuss a little bit about the autoimmune disease. This is actually loss tolerance to our own cell. Okay, so our own cell is become is considered as foreign cell by our own immune system. So the disease that caused by this problem is called the autoimmune disease. Again, mostly this is type 3. Right? Type 3 hypersensitivity, which is the one that is caused by the IgG, okay? The overreaction of IgG. Okay, so mostly type 3. Several type of autoimmune disease like lupus, eh, RA, Myasthenia Gravis, MS, multiple sclerosis, type 1 diabetes. This is also autoimmune disease. And this is example of systemic autoimmune disease like lupus, yes, systemic lupus erythematosus. This is maybe how it look like someone that have this type of autoimmune disease. Another one is RA, rheumatoid arthritis. This is on the joint tissue. So the joint tissue actually being destroyed by our own IgG. Endocrine autoimmune disease, like a Graft disease. This is also known as the hyperthyroidism, okay, which is uh, too much thyroid hormones. Hashimoto's thyroiditis, this is also known as hypothyroidism, less thyroid hormones produced. Type 1 diabetes mellitus, this is due to the antibodies that damage the beta cell in pancreas, which is the one that produces the insulin. Okay, so when the cell that produces insulin being broken, then there will be no insulin production, and it is caused by our own IgG antibody. Another autoimmune disease is called the neuromuscular, okay, like Myasthenia Gravis. So the muscle actually become very weak because of this disease. Okay, so muscle weakness, MS, multiple sclerosis, maybe some paralyzing in some muscles. Okay, muscular weakness, uh, difficulty to speak, vision, problem with the vision. This is a MS. Now I just wanted to show you how this disease occurs, okay? Why. Like for example, if we give examples, diabetes 1, type 1 diabetes. This antibodies over here, which is the IgG, is actually destroying the beta cell. So this IgG consider this beta cell as a foreign cell. Okay? So it will attack this foreign cell. Remember, antibody will destroy this antigen. So it's considered as antigens over here, which is actually not. Okay, so this is loss of the let me see over here. Okay, loss of tolerance to our own cell. Okay, so our own cell is considered as a foreign cell by the IgG. So this antibody actually attack our own cell and destroy this cell. So in this case, the pancreatic cells over here that being destroyed by the IgG. So the beta cell of pancreas being destroyed. Therefore, because the beta cell is the one that produces insulin, there will be no insulin production. Okay? And you know the type 1 diabetes, it is progressive disease. For example, maybe during the younger age, that person do not realize that his beta cell is being destroyed, okay? And then after several years, maybe 12 years or 15 years old, he start to develop this diabetes. Okay? Within 20, 25, 30 years old, then he become uh, dependent to insulin injection. Okay? So sometimes some people wonder why, when you're a baby, my my kid doesn't have the diabetes, and after 12 years old, and he developed the diabetes. This is the problem because this distraction take time, okay? So take time. After several years, then it's become the sign and symptoms will occur. Okay, so he will be dependent on the insulin injection after that. This is Myasthenia Gravis. It's the same thing, which is these antibodies, which is the IgG over here, is actually block or attack the acetylcholine. So normally acetylcholine will bind to the muscle cell, and then the muscle cell will be able to do contraction over here. Now, if the IgG over here actually attacking the acetylcholine, then there will be no contraction. So the cell, the muscle become paralyzed. So this IgG is actually block this receptor from getting the acetylcholine. So the muscle become weak, weaker muscle. So people that have myasthenia gravis, they usually have weaker muscle or weak muscle during movement, during contraction. I think that's all for chapter 16, and you will have the exam number 4 of chapter 14, 15, and 16. So good luck with your exam. Goodbye now.